So I decided to do my project on the technique of viral gene therapy. I think this is a very advanced field that can be, still expanded upon. So I was interested to do a little bit of research into it. So I'll go ahead and get started. I'll put myself back in the corner here, and we'll start to go through it. So the goals for today is to talk about what exactly is viral gene therapy, what is heart disease, as well as the procedure that we use, including the exercise study, gene therapy, and the insertion of that gene into cells, as well as the results of the study and conclusions based on that. So what is viral gene therapy? Viral gene therapy is the use of a viral vector to reintroduce genes into cells that may have lost these proteins that are being coded for, or they are so diminished that there is some sort of disease attached. This can be compared to when you order a package, which would be the DNA, and it is transported via some sort of mail truck, whether it's FedEx, UPS, or the post office. The vector itself doesn't matter as long as it gets to the cells. So we'll kind of talk about the different types there and how this kind of ties into that analogy. So we'll just go through a quick video on heart disease as well as some of the treatment methods that are possible for this before we get into the viral gene therapy.
Your heart is a muscle that pumps blood around your body. The term heart failure can sound frightening, but it simply means that your heart can't pump blood as well as it should. Heart failure can be caused by a number of things, most commonly a heart attack, high blood pressure, problems with heart valves, or cardiomyopathy, a disease of the heart muscle. Joe has just been diagnosed with heart failure. Because his heart can't pump blood around his body as well as it should, it's causing symptoms in different parts of his body. When blood isn't flowing properly, it can lead to a buildup of fluid or congestion, a bit like a traffic jam. This fluid sometimes builds up in Joe's lungs, leaving him breathless. Fluid can also build up elsewhere in Joe's body. He notices this as weight gain and swelling in his feet, legs, lower back, and abdomen. Joe also suffers from fatigue because his heart can't deliver enough oxygen-rich blood to his muscles. With your continued support, we're funding research to one day find a cure for heart failure. Right now, Joe uses medication to manage the symptoms and help him have a good quality of life. Joe has also made some lifestyle changes to help keep his heart healthy, such as quitting smoking, cutting down on alcohol and salt, staying active, and keeping a healthy weight. Joe doesn't need any further treatments at the moment, but because heart failure can get worse over time, he may need them in the future. Some people are recommended to have a pacemaker or implantable cardioverter defibrillator, ICD, to help treat their heart failure.
So this video not only shows the basics of how the heart normally works, it also shows you cardiomyopathy, which is that weakening and expansion of the heart wall that can make it hard to pump blood throughout the body. And it also discusses several treatment methods. So right now it says that Joe is using medication successfully, but surgery might also be a possibility. So in my opinion, I believe that before moving on to any sort of, this gene therapy that we're discussing, that maybe medication or a simple surgery that most doctors already know in practice might be a better first bet. And then if for some reason it got to the point where that person needed some sort of heart transplant, maybe a viral gene therapy option might be better before we get to that point. However, because it's kind of new and upcoming at this point, I wouldn't recommend it as the first course of action, but we're still going to go ahead and discuss it here today. So the target that we're using for this viral gene therapy is called the SERCA2A, and it is involved in muscle contraction. So the contraction of the heart muscle and the relaxation in order to make blood pump throughout the body. So how it works is that calcium enters into the cell as well as the sarcoplasmic reticulum, which is that steak-like pink thing you see there. It also releases calcium. So there's an excess amount of calcium coming in, and it causes muscle contraction. Then once the muscle is contracted, the white triangle you see there, which is PLN, which is an inhibitor, it releases SERCA2A, so it's no longer inhibiting it, it's letting it work. And then that allows calcium to be taken back into the sarcoplasmic reticulum and relaxation to occur. If SERCA2A is not present or present in very diminished amounts, then you're not going to have effective relaxation, thus making it so when the heart tries to pump, it's not as effective. In this study, we also use what were called knockout mice. So we have our wild type mouse here. It's normal. It's showing these green genes here that they're working. However, in our, not in our knocked out mouse, what you see is that these black arrows here show an inhibitor. And when the inhibitor is added, it makes it so anything after the inhibitor is not coded for. This means that, you can basically take out a gene, while 10 to 15% of these knockout mice are actually found to have a lethal mutation, so they don't end up being born alive to grow and study. These have been used in cancer studies as well as the one we're using here today for the SERCA2A mouse. Just a quick safety point, there'll be a few of these throughout the study. Proper PPE was always used, including gloves and lab coats, and then they didn't explicitly say to put on goggles, but they were available in the lab. So we did an exercise study. This was to confirm what was found in an in vitro study we were given in the background. So in the in vitro study, it was found that the SERCA2A could have an effect on heart disease. If it, we found decreased SERCA2A, it may have an effect on, an increased risk of heart disease. So what we did is the brown mice you see had induced heart failure. They were not knockout mice. They just had heart disease. And then the white mouse was a knockout mouse. It was a SERCA2A knocked out. This could be why it's white in color, because you can sometimes see a phenotypic change, but that wasn't said for certain. One of each of these mice was placed onto the treadmill on your right at their own respective time, and the intensity was placed to high. Both mice did die during this exercise, and that can help to show that the SERCA2A protein and lack of it, so lack of that gene, lack of the protein, may actually be a cause of heart disease due to the fact that they both showed the same result, but this can't be confirmed until we continue the study. So what exactly are we going to do to try to treat this? You're going to take that therapeutic gene, put it in your viral vector, and then put them into the heart to try to make it so you can help them to produce either, start producing or produce more of this SERCA2A gene to then make more proteins so that you can effectively reduce the risk or even cure entirely their heart disease. Just a point of safety, what if the viruses are spread outside of the heart? Could there be further adverse side effects? The answer is no. The viruses use what's called an alpha myosin heavy chain promoter, which means that if it's outside of the heart muscle, it's actually not going to have any effect. They're not going to be able to reproduce. So they're only going to be effective inside the heart. Other viral vectors include the, so again, we talked about the AAV. We'll talk about that in a minute. We also talked about adenoviruses, which are on your right. And we also have the retrovirus, which is in the center. You may have heard of all of these. Others also include the vaccinia and the polio vectors. The one that you might most commonly know is the retrovirus. This goes against the central dogma and can kind of, transpose the actual RNA back to DNA, can just change it in that sense. And this has been shown to be a very effective method. However, it has also been stated that if there is some sort of back mutation, so it doesn't work properly, it can actually introduce an oncogene, or it can suppress a tumor suppressor gene, meaning that it could cause cancer. So that is why we're actually looking to use this AAV, or adeno-associated viral vector, because it has no pathological effect to humans, unlike most of the other vectors that were discussed. It can also be just as effective as these vectors. The only issue is it has a small size, so it has a small carrying capacity of about 4.8 kilobase pairs. It can also be expensive to create. It may require a helper virus, so we'll see that later. And previous AAV therapy could cause antibody neutralization, so it could cause an antibody response that could be picked up if this were to be injected for a second time. However, that's just a speculation from the researchers that hasn't actually been tested. So there are actually 3 plasmids that are needed to produce an AAV. So there's the gene of interest, which in our case was the SERCA2A plasmid. Note that that's yellow with red tips. You'll see that later. The viral plasmid that has the rep or replication region that allows the actual plasmid to be reproduced, and the cap region, which encompasses the genes needed to make a capsid, so make that icosahedral 20-sided, kind of like a 20-sided dice that you would see. That protective layer is made by the cap region. And then the helper plasmid, which in our case, they didn't specifically say, but it could be something like an adenovirus. This will help in the yield overall. However, it wouldn't be required, but it was used for this case. So just another safety point. Again, everything we did when it came to the viruses was done in a biosafety hood. This is different from a vent hood because while it does protect the researcher from inhaling anything, so in this case, inhaling viral or bacterial pathogens instead of just chemical fumes, it also creates a sterile environment due to a HEPA filter. So all of the air coming in contact with your sample is completely sterile to reduce the risk of contamination. So the way these were produced, there were two separate tubes. So tube A, which is the orange cap, had all three of the plasmids as well as cell culture medium to keep them viable. And cell B had a transfection reagent as well as, again, cell culture medium. These two tubes were then combined, vortexed, and left to incubate. It should be noted that it took multiple steps to kind of fill these tubes and then another step to transfer them. And then after incubation, it takes another step to put them on a cell culture plate. These multiple transfers could lead to some error. I don't think it would be a great error, but it may be enough to cause some sort of change, so it should be noted. Also, again, once it gets onto the plate, it's put onto HEK293 cells. We'll talk about those in a minute. It is allowed to incubate on the plate for 2 days at body temperature. And then these are added to mice. In this case, it was one mouse. And they are incubated within the mouse for 5 weeks. So why are we using these HEK293 cells? They're long-lasting, so one cell line can actually last up to 10 years. They're very permanent cell lines, so they're very stable and usable in this case, but they are difficult to obtain. The HEK actually stands for human embryonic kidney cells. So they're coming from human embryonic kidneys, which seem like they'd be a little hard to obtain. And then you also have to take these cells, and they're actually genetically modified, you could say, with an adenovirus. So they're pretty complex, but again, they do last for a long time. So if you do get a cell line, it could last for up to 10 years. So this is just a quick snapshot of what's going on. So the bumps you see on the left are actually the cells. So the cells are taking in that viral capsid. Again, it looks like that dice shape, that 20-sided dice shape. Top right, you're seeing those viruses again zoomed in. And then on the bottom left, you're seeing the capsids begin to break apart. So on the bottom right, they can release that DNA. Again, we know it's the circa DNA because it is the yellow with the red tips on it. So how do we know that only the desired gene is being packaged? How do we know it's not a contaminant? Well, they have what's called inverted terminal repeats. Those are actually the red end tags that you see. So they make it so if these repeats are not present, they're actually not packaged. So it's kind of an extra check and balance mechanism. So once we allowed them again to be in the mice, or mouse in this case, for 5 weeks, they were sent through another exercise trial, again on high intensity, and this mouse actually survived that trial, which was a good sign. In order to get a numerical value, instead of just saying, oh, the mice survived, the mice died, instead we put numbers to it, so it's an ejection fraction. This measures the amount of blood that's pumped out versus how much was fully there. So it's a percentage of basically, how effective is the actual, ejection of blood from the heart. It was found in the control mouse that it was about 70%. Mouse with induced failure showed that it was 30%. The mouse with induced heart failure and just the vector was also 30%, showing that it wasn't the vector that caused this change. There's a nice control in there showing that it wasn't the vector, it was what we see in the next one, which is the vector with the SERCA2A gene. And that increased the ejection factor to 60%. So that shows that it's actually that SERCA2A that's showing the change instead of just the viral vector. But what do these numbers actually mean? So again, this is human data from the American Heart Association. They say that a normal ejection fraction is from 50 to 70%. So that means our control mouse, as well as our mouse who has received the treatment, are both within the normal range. If the ejection fraction is less than 40%, so what we see in our mice with induced heart failure and the one with induced heart failure and just the vector, they are said to have heart failure. But, however, with the treatment, again, we're going from heart failure up into the normal range. So while it's not pristine and back at 70%, you could technically say that we have treated their heart failure and brought them back to a normal level. So just an overview again, that heart disease or heart failure treatments are available, and that viral genome therapy is another option, again, until this becomes well established and really is accepted by a lot of doctors so they're comfortable with it. I wouldn't say it should be the first line of treatment. However, it is an option. AAV is the best choice for this experiment. Again, while it is small, we didn't really need a big carrying capacity, and the fact that it's not pathogenic for me is the biggest draw to this specific vector. We also were able to target the SERCA2A gene. We knew this from previous in vitro studies, so we weren't just, picking a random gene and trying to see if we can make the mice, fix it based on what we thought. We had previous studies and the exercise study to confirm that. We also used a well-established procedure. Not only has this, again, been used in cancer trials, but it's also been used in trials for Huntington's and Parkinson's disease, so we know it's a valid method. And we also did see a positive result in the mice. So again, we did only see one mouse for each value, so I would have liked to see a repeat in the amount of trials. However, I think this is something that maybe could be expanded on, put into human studies, and really looked into further. So again, I believe that while it may be a little expensive to kind of start up with this procedure, if you do have a lab who is equipped to handle this and is willing to take on the project, I think this would be a very effective form of treatment, and may eventually one day actually, take out some of those other treatment methods that have been around for a while, if we can kind of get it hammered down and really figure out what the best form of attack for it is. So again, I really recommend this procedure.